So chapter 9, we put these two chapters together. Chapter 9 is all about controlling microbial growth out in the environment. So various types of ways you can clean and kill, and whether it's using heat or temperature or cold, whatever it is, all in the environment. Chapter 10 is all about controlling microbial growth in the body. Obviously we can't use incinerators, we can't freeze them out, we can't use salt, things like that. So these are the different types of drugs that you can take and how the different drugs work at killing the various organisms. So the chapter 10 part of the PowerPoint is we're going to go over some terms we're talking about when we're talking about antimicrobial drugs. What's the different mechanisms of actions, meaning how do these different drugs work? What are some considerations when we choose a particular antimicrobial drug? And the unfortunate of what's happening these days is that resistance to antimicrobial drugs. So first, starting with some terms, and again, you can pause anywhere in here. I wrote in everything as well, so it's a nice place you can pause. But drugs, anytime we talk about a drug, it's just chemicals that affect physiology in any manner. So whether it's illegal drugs, over-the-counter drugs, all of these things, these are chemicals that affect physiology in the body in some form. A chemotherapeutic agent, these are different types of drugs that act against diseases. Now, that doesn't even mean microbial diseases. This could be if you've got any, cancer, or you've got some type of depression or something. These are just any other type of drug that treats the body, treats some type of disease, treats some type of disorder. What we care about in our particular course are the antimicrobial drugs or antimicrobial agents. These are drugs that are treating particular infections, meaning something that got in the body that's not supposed to be, and that's what these drugs are working against. Now, 3 groups of antimicrobial agents. Antibiotics are naturally produced antimicrobial agents, meaning these are found out in nature. And I'm like that, yes, there are different types of fungi that can inhibit bacterial growth. That's where we got penicillin from. These are naturally produced antibiotics. Semi-synthetics, these are antibiotics, but we chemically alter them some form, whether it's to make them last longer, more effective, easier to administer. And I'm like, we've changed them a little bit. And synthetics are completely synthesized in a lab. These are not anything found out in nature but tweaked. These are completely man-made antimicrobial agents. Now, when we're talking about different types of antimicrobials, we also are talking about that they are selectively toxic, we hope, meaning they're more harmful to the microbe than they are to the patient. We would not want them to hurt the patient. The idea is to help the patient. And of the different antimicrobials we have, antibacterial drugs are the largest number of antimicrobials. We have a lot of antibacterial drugs out there. Eukaryotic infection drugs, limited supply, and antivirals, we have limited availability. We have some drugs on the market to treat some viruses. I've got the picture of the Tamiflu for one, and we have some drugs to treat various eukaryotic infections, different types of parasitic worms and things like that, but limited availability. Now, when we are choosing what type of antimicrobial drug, granted, you guys probably aren't going to choose, but you're going to, help administer. But when a certain drug is chosen, they are looking is how is that antimicrobial going to work? How is it going to kill the organism or inhibit that microbe? These are the different types of mechanism of actions. It's like, how does it kill? How does it work? And yes, there's lots of mechanisms of actions. And I'm like, these drugs all inhibit cell walls. And again, each one of these are going to be my next slides. We have a whole group of drugs or a class of drugs that inhibit cell walls. We have a whole class of drugs that inhibits protein synthesis, a whole class of drugs that disrupts the cell membrane somehow. We have a whole class of drugs that inhibit various metabolic pathways. We have a class of drugs that inhibits the making of DNA and RNA, and we have a class of drugs that inhibit organisms from attaching or recognizing a host. So these are all the different classes of drugs or the mechanisms of actions. How do they work? How do they kill these microbes? We're going to start at the top and work our way around like a clock. And yes, out of the different classes, You may actually recognize the names of these antimicrobials the most in this class. This is the most common mechanism of action. So you're like, oh, I recognize some of those. Most common mechanism of action on antimicrobial drugs. And I'm like, is inhibiting bacterial wall synthesis. Again, all bacteria have some type of cell wall, whether it's big, whether it's thin, whether it's thick, but they all have some cell wall. And the basic idea of how these drugs work is they prevent the peptidoglycan formation. They prevent the bacterial cell wall from forming. Now, we call this class of drugs a beta-lactams class of drugs. And I'm like, the beta-lactams itself prevents NAMs and NAGs from attaching to each other. Now, if you remember, and I'm like, all the NAMs and NAGs, we're going to call them, my peach and orange here. And I'm like, they all attach to each other, side to side, from one chain of them to another chain. They're all attaching to each other. Well, what this class of drugs does is it prevents these little crosslinks. It prevents these attachments. And if they can't attach and form this nice, solid, strong wall, you're going to have a really weak wall. And what ends up happening is it's a weak wall that water pressure on the inside or the outside can cause the bacterial cell wall to fall apart, to burst open, to, rupture. And I'm like, and if you've got a weak wall, the bacterial cell will die. Yay, because that's what we want to do. Now, just some examples again, and I'm like, there's a bunch on the list: penicillin, cephalosporins, bombactins, and I'm like, most common, and I'm like, antibiotics given. Other types of antibiotics that are given, these are these are the natural antibiotics. The ones that are found out in nature are semi-synthetic derivatives: methicillin, amoxicillin, ampicillin. It's pretty much taking penicillin and then tweaking it a little bit. Now, people that have allergies to penicillin a lot of times have allergies to semi-synthetics as well. Not always, though. Other types of drugs that inhibit bacterial cell walls, some are a little more specific or in how they work. Vancomycin and cycloserine both work against the gram-positive bacteria cell walls, inhibiting their formation, but they're more effective against gram-positives. Bacitracin blocks NAMs and NAGs, not from cross-linking, but from ever leaving the cytoplasm to form. So they still work against cell walls, just a slightly different way. And the isoniazid and ethambutol both disrupt the mycolic acid formation. These are the ones, the mycobacterium, the ones that have to have acid fast stains done to them. That's that lipid in their cell walls. So they don't work against the NAMs and NAGs, they work against the mycolic acid formation. Another mechanism of action is inhibiting protein synthesis. Now, making a protein goes through two big steps. It goes through transcription and it goes through translation. Well, most of these drugs are going to target the translation step. They're going to target that messenger RNA from being read in the ribosome and forming proteins. And again, if you don't have proteins, you're not going to survive, because all of your enzymes are proteins. All your structure materials, proteins. Everything needs proteins. If we can stop the protein formation, even in prokaryotic cells, we're going to stop them from living. Now, different drugs work in different ways on stopping translation. Again, ribosomes read messenger RNA, and they're going to put amino acids together and form proteins. Well, different ways they can do. One, they can change the shape so that it's not read correctly. They can change the shape of the ribosome, and it won't read the messenger RNA correctly. Some, they can affect somehow in the ribosome or affect the messenger RNA that your transfer RNAs can't read it correctly, and if they can't read it, they can't, attach, they can't bring in their amino acid to string them together. Some drugs stop amino acids from attaching to each other. So if you can't attach amino acids together, you're never going to make a protein. Some stop the messenger RNA from getting pulled through. If it can't get pulled through, it can't get read, can't assemble. Some stop ribosomes from even attaching to the messenger RNA. And I'm like, whether either side or whether one of the parts of the RNA. Either way, if you stop translation, you stop proteins, microbes not going to survive. Another mechanism of action is somehow disrupting that cell membrane, that cytoplasmic membrane. One of the best ways to do it is form a channel or a hole where there's not supposed to be a channel or a hole. That's going to disrupt the integrity of that cell membrane and how molecules go in and out of the cell. Again, those channels, the cell membrane, keeps things in it wants in and keeps things out that it wants out. If you have holes or channels that aren't supposed to be there, things are going to get out that aren't supposed to, and things are going to get in that aren't supposed to. One of our top drugs that disrupts the cytoplasmic membrane is amphotericin B. Works against fungal cells, so this is for fungal infections. How it works is that it recognizes in the cell membrane of a fungus, they have these purple molecules called ergosterol. And anywhere the drug sees ergosterol, it's going to insert a pore or a channel. Again, disrupting integrity and molecule flow. And I'm like, and it's extremely effective against fungi. It's our top drug against fungal infections. So on our one PowerPoint later this semester, you're going to hear me say the word amphotericin B a lot. Now, problem with humans though is our cells have a similar looking molecule also in our cell membranes called cholesterol. And I'm like, that sometimes amphotericin B recognizes cholesterol as ergosterol and it inserts pores or holes in our cell membranes. So amphotericin B is great at killing fungus. However, it causes a lot of side effects in humans. None of them are good. So I'm like, it's a great antifungal, it's our top antifungal, but it generally causes lots of side effects. Bacteria, not affected because bacteria don't have any type of ergosterol or cholesterol. So it is, working for fungi with side effects. Another drug that works against the cell membranes, but it does work against bacteria, not fungus, is the polymyxin problem. It works against gram-negative cell membranes, and they have two. Problem as it is, it does cause some toxic side effect to human kidneys. They're hard to treat. So generally, if you've got different types of bacteria you're killing, you're not generally going to go with one that disrupts the cell membrane. Another mechanism of action is to inhibit some type of metabolic pathway. The metabolic pathway is where an organism can take one substrate, use an enzyme, turn it into another substrate, use an enzyme, turn it into another substrate, and eventually make an end product. That's the pathway. One substrate to an end product. Well, the drugs are going to inhibit a step somewhere along this metabolic pathway, whether it's inhibiting an enzyme here or somehow disrupting one of the substrate. And I'm like, but it's going to inhibit one of these. We just have to make sure that the anti-metabolic agent works against a microbe's pathway and that our cells don't have a similar pathway or a similar enzyme, or it's going to disrupt that. Couple examples of some of these drugs: adaviquone interferes with electron transport needed to make ATP. Some heavy metals inactivate enzymes. Just have to make sure it's an enzyme that these microbes have and not ours. Or we've got drugs that block the activation of viruses, that there is a pathway to activate a virus so it starts to reproduce. If we can stop somewhere along the pathway, viruses aren't activated, they're not going to reproduce and kill cells. We've got some antiviral agents. These affect some type of viral metabolism, how they reproduce. And we also have protease inhibitors. This is a particular drug that works specifically against the enzyme protease. Again, most enzymes end in ase. Now, the only main organism that has the enzyme protease that it needs is HIV. And I'm like, so it's a very specific inhibitor that inhibits that particular enzyme that's needed in the HIV metabolic pathway. Another mechanism of action are drugs that inhibit nucleic acid synthesis. A lot of stuff on this slide. And I'm like, the main part is that, yes, and I'm like, cells need to make new DNA, lots of enzymes along the way. Whether it's new DNA or new RNA, if you disrupt somewhere along that, it's a type of metabolic pathway, but it's disrupting a specific metabolic pathway to making DNA and RNA. So it's disrupting somewhere that at some point you're not going to make DNA or RNA. Now, whether it's blocking some type of transcription, making DNA or RNA and transcription, And I'm like, it's usually used, and I'm like, it's not usually, it's used against both eukaryotic and prokaryotic cells. Either one, they have DNA, they have RNA, there's lots of ways we can stop it. Our only little thing is, because we are eukaryotic and we have DNA and we need DNA, there can be lots of possible side effects. So right now they're still doing a lot of research on drugs that inhibit nucleic acid synthesis. So it's mostly used in research, and it is used to slow some cancer cells as well. Again, for cancer cells to divide and grow, they need to make DNA and RNA, just like our cells do. Well, if we can stop that, we can stop or slow down cancer cells. Now, how it does that, like without going into lots of details, a lot of these use what are known as nucleotide or nucleoside analogs. Again, we've got 4 different nucleotides in our DNAs, our A's, T's, C's, and G's. Well, these are analogs or lookalikes, A's, T's, C's, and G's. So these are organisms or molecules that look enough alike, these nucleotides, that when we try to assemble, or when the microbes try to assemble these A's, T's, C's, and G's, they may bring these lookalikes in instead. And because they're lookalikes, even just one small little part, the DNA is not read correctly. It's incorrect DNA. So it's almost like we're tricking DNA synthesis by having these lookalikes, A's, T's, C's, and G's. Now, some specific drugs, the quinolones and fluoroquinolones, work against bacterial DNA gyrase. It's a particular enzyme in making DNA, found specifically in prokaryotes, and these drugs work against the stopping of that one particular enzyme. Otherwise, there are drugs that work against RNA polymerase, the enzyme that's used in making RNase, and we have drugs that work against HIV. Again, another enzyme. You notice these all end in ACEs, they're all enzymes. And there's a very specific enzyme that HIV uses called reverse transcriptase. It does reverse transcription. Only HIV does reverse transcription, so these are drugs, if they can inhibit that one particular enzyme, it's very specific, low side effects for us because we don't have that enzyme, we don't ever do reverse transcription, so it's really affecting just that one particular microbe of HIV. Now, note, as I've mentioned HIV a few times, we've put a lot of research, aka a lot of money and dollars into researching HIV, so we do have some drugs and benefits of all that money and research. And then another mechanism of action is preventing microbes from recognizing and attaching to cells. Before a virus can get inside of a cell and hijack it, it first has to attach to it. If we can prevent a virus from attaching to a cell, it's never going to get inside of it, which means you're never going to have a viral infection. It's our newest area of drug development. I hope it works. And I'm like, we just are trying to prevent them from attaching. Now, those are different ways they work. Some considerations when we're trying to figure out which antibacterial drug is prescribed. One, our best ideal antimicrobial agent, this would be, gold standard, everything we want from a drug. We want it readily available. We don't like to wait. We want it cheap, always. We want it chemically stable. We don't want this thing blowing up on a shelf. We want it easily administered. We want it non-toxic, non-allergenic. We want it selectively toxic against a wide range of pathogens. It means we want it toxic to a lot of things, but selectively toxic so it doesn't hurt your patient. And we want it effective against your patient's particular bacteria, the particular strain of bacteria or microbe that your patient has. That's our ideal antimicrobial agent. We ask a lot of our drugs, but we have a lot of drugs that we've been able to do this for. Now, 2 kind of groups of drugs that we do have when we're prescribing these different antimicrobial agents. One, we have narrow spectrum drugs and we have broad spectrum drugs. And when we're talking of a spectrum of action, we're really talking about how many pathogens a drug can act against. These drugs, other than some of the ones for HIV, aren't generally very specific for killing one and only one organism. Some of these drugs will kill a multitude of organisms. That's the spectrum of action. How many will these kill? A narrow spectrum drug is effective against a few microbes. If it's HIV, some of these drugs are specific against one. That's a narrow spectrum. Then we have broad spectrum antimicrobials, which means these are drugs that are effective or can kill many microbes or many organisms. And a problem with broad spectrum antibiotics, I'd say, well, we'll go the good and then the bad. The good with a broad spectrum antibiotic, you don't even have to ID out a very specific microbe in your patient. If a patient comes in and you're like, oh, they have an ear infection or a sinus infection, as many of you have been diagnosed with an ear infection or a sinus infection, do they know exactly what bacteria it is that's caused that ear infection or sinus infection? Because they're not all the same bacteria that can cause those. You just know you have some organism that's gotten in your sinuses, there's some organism that's gotten in your ear. You don't have to ID it out. You treat it with a broad spectrum antibiotic and it kills a lot of microbes and it's going to kill the one that got up in your sinuses and your nose. That's the good thing. You don't even have to fully ID it. You're just given a broad spectrum antibiotic. The problem with broad spectrum antibiotics is that because they kill a lot of different organisms, they kill your good bacteria too. Not all bacteria are bad. You've got good bacteria, and broad spectrum antibiotics are going to kill your good bacteria, your normal flora bacteria. And what ends up happening is you get these secondary infections because you killed your good bacteria. Now, these are just looking at a couple of antibiotics that you may recognize. Erythromycin, it kills almost all gram-negatives, all gram-positives, and some of our unusual bacteria called our chlamydias and the rickettsias. It's a broad spectrum. All gram-positive, almost all gram-negatives. Again, you don't have to ID out the particular bacteria. Just give them erythromycin or tetracycline and you're going to kill it. And I'm like, and killing good ones as well. Other than, how many do we want to kill? We also do want to make sure that the drug that's given is the most effective drug that can be given. And there are some tests that we can do to make sure that the drug is effective against your patient's bacteria. So we want to know its effectiveness. One, we can do a minimum inhibitory concentration test or a minimum bactericidal. Inhibitory, it just inhibits bacteria growth. Bactericidal means it kills the bacteria. We want to know what's the minimum amount of drug that's either going to inhibit bacteria growth or going to kill the bacteria. So similar tests. Now, we're going to do this particular test. We're going to use a Kirby-Bauer plate, and our Kirby-Bauer test, and we're going to cover a whole plate with an antibiotic or with a bacteria, and we're going to put a couple antibacterial soaked discs on the plate. And we're going to look to see, and I'm like, which antibiotic kills or inhibits the bacteria the most. I'm like, this particular antibiotic, whatever it is, has no bacteria growing around it. That's a pretty effective antibiotic. These antibiotics, the bacteria grow right up to the antibiotic. They could care less. It's probably not an antibiotic that you want to give. And so I'm like, we can look at different antibiotics to see which ones are effective. And we could also use these and test them. These could all be the same antibiotic with different concentrations, and we want to see which one is most effective. Although we could do that and have different concentrations on each one of these discs, that is usually used for an E-test. And I'm like, the E, and I'm like, is just testing. It's a strip of paper that all along the strip has various concentrations of the antibiotic. Strongest antibiotic at the top, and then each little number or little jot down is a decrease in concentration of antibiotic. And we're really looking, what is the lowest concentration of antibiotic that has some effect on the bacteria? Only because we don't want to have to overprescribe an antibiotic. You don't want to have to give a patient 256 times the amount of antibiotic that's needed. And I'm like, it's too much antibiotic. It might actually cause more harm and more side effects. So you're just looking for, what's the minimum amount of antibiotic that's needed to cause that inhibition. Now, we also want to know how are you going to prescribe? You're going to prescribe an antibiotic, but how is it going to be given? What are the routes of administration? And I'm like, is it going to be a topical application? Because it's just an external infection. I'm like, you can see this infection on the skin. You can do a topical application of antibiotic. Is it going to be given orally? Is it going to be given intramuscular through some type of injection? Or is it going to be given intravenous, right through the bloodstream? Now, comparing oral versus intramuscular versus intravenous. The red line is looking at how much of the drug gets into your blood, and I'm like, in a particular time frame. And comparing the three, oral, intramuscular, intravenous, which one gets into your bloodstream with the highest concentration and the fastest? Well, it's the intravenous, because you are giving that drug right into your blood supply. You're going to get the highest amount into your blood supply the fastest. Intramuscular, about half of the drug is going to make it into your blood supply, and it's going to make it, almost as fast as intravenously. Oral, not as much gets in, maybe a third gets into your blood supply, and it's going to take even longer. Now, although oral gets into your blood supply the slowest with the least quantity, it is the number one way most antibacterial drugs are administered. Reason why? It's the easiest and the cheapest. And I'm like, if you're given an antibacterial drug, it's almost always given orally, is take this pill twice a day for the next 14 days. If you were given intramuscular, either they're going to send you home with a whole bunch of shots that no one's going to want to give themselves into the muscle, or you're going into the doctor twice a day for a shot for 14 days. No one wants to do that. It costs a lot. Same thing with continuous intravenous. But if a patient does have a very severe infection of bacteria that they need to treat and kill those bacteria as fast as possible, you're going to go with it intravenous. You're going to want that high amount of antibiotic in that patient's blood as fast as possible. If they're not as worried, oral, send you home. We also want to know, for the drugs that are given, we want to make sure it's not toxic to any of the patient's organs. Kidneys and the liver are the top two organs that show toxic side effects. Again, it's because of all that blood that goes to those particular organs. We also want to know if there's going to be any toxic side effects against a fetus if your patient's pregnant. You also want to know if there's any possibility of allergies in your patient. It's rare to have antimicrobial allergies. However, it's possible. There are quite a few people allergic to penicillin and amoxicillin. And I'm like, fatal allergies? That's really where the rare comes in. And we do also want to know, how will it affect those normal flora, your good bacteria, because it results in other infections, and that is a huge concern when you're prescribing antimicrobial drugs. One of the top secondary infection concerns is C. diff. C. diff comes from killing your normal flora. Your normal flora bacteria, your good bacteria, keeps C. diff in check. You kill them, those C. diff infections just grow like crazy. And then our biggest concern these days, because we have all these antimicrobial drugs, is our bacteria are becoming resistant to the antimicrobial drugs we have. Now, some bacteria are naturally resistant to certain antibiotics. Staphylococcus aureus, the main staph infections, it has enzymes that can break apart certain antibiotics. It naturally has that, so they're naturally resistant to certain drugs. What we're concerned more about is that a lot of them are starting to develop bacterial resistant. Some of it's through natural selection, that they get a mutation, and that mutation is beneficial. That's rare. Most mutations are bad. And I'm like, but the more common way that bacteria are developing resistance is through genetic recombination, aka they're sharing genes, they're sharing DNA, and that comes from plasmids. Plasmids, we'll talk about them later, and I'm like, they're little snippets of DNA that bacteria have. They're not needed to survive, but because they are DNA, some of that DNA can code for antibacterial resistance. In the video Hunting the Nightmare Bacteria, when they talk about NDM1, That's a gene. That's DNA that codes for antibacterial resistance. How they get that plasmid, how they get that snippet of DNA, transformation, meaning they can literally get absorbed into the bacteria, but the most common way is the conjugation pili, when different bacteria connect to each other to exchange genetic information. Now, how the bacteria, become resistant. We know they can have these genes, but how do those genes become resistant? We've got mechanisms of action, how the drugs work, but then there's mechanisms of resistance, how these bacteria are resistant to these drugs. One, some of these bacteria will make an enzyme that destroys or deactivates the drug, so the drug can't do anything. Some microbes slow or prevent the entry of the drug into the cell. Gram-negatives are great at that. They've got 2-cell membranes to pump drugs out. And I'm like, drug can't get in, drug can't do anything. Some alter the drug, so they change the drug so it can't bind to the cells or bind to its target that it's supposed to kill or get rid of. Some will alter their metabolic chemistry. And I'm like, meaning they're going to change their pathways, their metabolic pathways. If you have a drug that inhibits an enzyme in some type of metabolic pathway, well, they'll figure a way around that. They'll make another enzyme and eventually get to their end product. Some pump the drug out before it can ever get in. It's, the drug gets in the cell and they just pump it right back out so it can't do anything. And some form biofilms, which biofilms themselves, because they form coverings and they have that protective matrix around them, they resist antimicrobial drugs from ever causing any type of action. Now, when we start talking about resistant drugs, when we start talking about superbugs, a superbug just means it's resistant to at least one class of drugs. So one whole group of drugs and how they work. Now, a lot of them have this resistance because they have some type of gene, some plasmid, like the NDM1 in the video, that codes for resistant to a particular class of drugs. Now, the problem with resistant drugs is that most of them are forming in hospitals and nursing homes, which is where you guys are going to mostly probably get jobs in your future. They're the breeding ground of resistant drugs. You have lots of microbes all hanging out together. That's who's in hospitals and nursing homes, sick people. They have lots of different types of bacteria that are all hanging out in close quarters with each other. And there's lots of antimicrobial drugs that are being prescribed. The more you expose drugs to bacteria, the more opportunity they have, ways to figure out a way around those drugs. I've got lots of little clips that come up here. You Google clip art for antimicrobial drugs, there's lots. When we talk about a multi-drug resistant pathogen, we're talking about these are organisms that are resistant to at least 3 drug classes, which are getting really scary. And cross-resistant or pan-resistant, as the video talked about it, means these are bacteria that are resistant to all antimicrobial drugs we have. It means there's absolutely nothing we can do for them. And I'm like, we hope their immune system gets rid of it or we have to cut out infections. There's not a whole lot. There's nothing else we can do. Now, our best way is to try to slow down this resistant. We can't stop it, but we can slow it down. And there are 2 top ways to slow down resistance. One is to maintain a high concentration of antibiotic. Means when you're prescribed an antimicrobial drug and they say take it for 14 days, twice a day, even though you're probably feeling great by day 3 or day 4, don't stop taking it. Maintain that high concentration twice a day for 14 days. Because the first bacteria that are killed are the weakest, which means the strong survive. And if you don't kill them all, those strong are going to do what they do best and they're going to reproduce. And strong bacteria make more strong bacteria. So maintain that high concentration. Kill them all. Kill all those microbes, even the strongest ones. Sometimes it takes up until day 14 to kill the strongest. But if you killed them all, they can't reproduce. There's nothing left to reproduce. Sometimes it takes a combination of antimicrobial drugs to kill them all. And I'm like, but you want to kill all of those bad bacteria so that they don't reproduce. The second is only use antimicrobials when necessary. The more you expose these antimicrobial drugs to bacteria or other microbes, the more opportunity they have to adjust to the drugs. The more ways, the more opportunities they can figure out a way around the drugs. And then we have to start coming up with new variations of drugs we have, or find new antibiotics, not likely, or come up with some type of semi-synthetic or synthetic drugs. And unfortunately, there's not a lot of money in developing new antimicrobial drugs. So it's, we don't want to expose them to anything that we don't need to, and we want to kill everything there is. Now, this link I have on Blackboard, so you can watch the video by yourself on Blackboard. But these are the top 2 ways to slow down antimicrobial drug resistance. And that's the end of this PowerPoint. It's, again, the whole PowerPoint is how do we kill the organism once they've gotten in the body.